Welcome to the Assay Engineers. In this video I show you how to use the Hyperplex biomarker configurator. Hyperplex protein profiling is a breakthrough, powerful technology that profiles more than 800 protein markers with a dynamic range of 5 logs from just 20 micrograms of sample. The assay menu currently includes more than 1,250 markers covering the top 10% of the intracellular proteome. You can choose from more than 60 expert-curated pathways, including FDA-approved companion diagnostic markers. The assay configurator currently includes more than 300 phospho-proteins that are not measurable by other platforms. If you are missing a marker relevant to your project, we can easily add custom markers and proprietary antibodies to your Hyperplex panel. Unlike other platforms, Hyperplex does not focus on discovering new, exploratory biomarker candidates. Instead, our platform makes validated biomarkers and a wide range of relevant pathway data easily accessible from a tiny sample. With this big picture, your biomarker decision can now be based on a comprehensive understanding of all relevant pathways. Our service lab provides you with actionable data to help you make an informed biomarker decision. And one more thing, Hyperplex is a targeted ligand binding assay platform. We can therefore provide you with the antibody information so that you can use the same clones and the same reagents in multiple programs, ensuring comparability and full transparency for your studies. Let's first download the Hyperplex biomarker configurator from the AssayEngineer website. Go to the intro sheet and activate the Excel sheet. Please make sure that the display settings of your monitor are compatible with the Visual Basic macros of the Excel sheet. This Excel file is basically a database of validated Hyperplex markers giving you an overview of information relevant to your biomarker selection such as antigen names, phosphorylation sites, gene information, etc. For proteins that are targets of FDA-approved therapies, you will find the drug names in the O column. Simply select the information that is relevant to your biomarker decision. Now we select the sample type. For example, we now select only those antibodies that are cross-reactive for human epitopes. Then we select groups of markers based on a disease area or pathway of interest, such as signaling panels, developmental pathways, immune signaling pathways and so on. For example, the database includes 567 human proteins involved in cell signaling, and 350 of these proteins and phospho-proteins have been shown to be involved in the mapped pathway. Take your time and select the signaling pathways you are interested in. In the final step, you can add disease-specific markers or pathway activity panels of interest, and you can even add markers that quantify approved drug targets. By the way, Hyperplex Assay Configurator is also a great tool to learn about expert-curated biomarkers for many diseases and many pathways. Finally, go to the F column and check the boxes marked with X and the configurator will display the markers you selected. Save the spreadsheet and email it to the Assay Engineers. We will provide you with a quote within 24 hours. The Hyperplex platform helps you quantify target interactions, pathway modulation and disease modulation. Does your drug candidate interact with the target? What are the downstream consequences of the drug-target interaction? Within 2 weeks of sample receipt, AssayEngineer's service laboratory will provide you with a comprehensive, actionable overview of the relevant activated markers and pathways. If you have any questions or would like to speak to a scientist, please contact us at hello@assayengineers.com.